Okay, guys, this is going to be our final couple of slides here on the integumentary system that we're going to get through in lecture today. I'm going to turn my video off, and now I will share my screen. So you should have my lecture in front of you now. Glands, we're going to talk about now the structures that we began discussing a little bit in our tissues lecture. Defined as a cell or organ that secretes substances for use elsewhere in the body or releasing them to be eliminated from the body, which is what we'll kind of focus on in this lecture. That is what we call exocrine glands. Endocrine glands are glands that we'll talk about later in the semester. They're the organs like our thyroid gland, pituitary gland, adrenal glands that secrete hormones directly into the bloodstream, and that's why the endocrine glands have a high density blood capillaries. Exocrine glands are like the sweat glands, oil glands, and our skin. Key features associated with those that have a duct, an epithelial tube, conveys their secretion to the surface. Also, examples of exocrine glands would be like salivary glands that secrete saliva into the interior mouth, various digestive glands in your stomach and intestines. There's a lot of exocrine glands throughout our body. Most of these exocrine glands are enclosed in a fibrous capsule, dividing the entire structure into smaller lobes and lobules, just kind of subunits and smaller subunits. The cells that actually perform the task of synthesizing and secreting are called parenchyma. Not super important stuff. Let's talk about the way that we categorize glands. Structurally, we can categorize them based on two criteria: whether their ducts are branched or not, so branched or unbranched, and then the shape of the secretory portion of the glands, which is what these looks like 8 pictures are kind of showing off. So there's some terms that go in to naming them, kind of like what we talked about with epithelial tissue. First term that we use will either be simple or compound. Okay? Simple glands have an unbranched duct, which in these pictures, the duct portion is kind of the purple, bluish shaded portion of our pictures. So if it has a branch or not. If it's not branched, it's got a name simple. If it's got a branched duct, we call it a compound. And then there's a second term, okay, and that's what tubular acinar and tubular acinar are. So if the duct and the secretory portion are of uniform diameter, the gland is called tubular. So if this width and this width are basically the same, we can call it a tubular. Now there's branched tubular versus just tubular, same thing. It's just, there's multiple portions to the secreting portion. Okay. If it's a little bit fatter in the secreting portion versus the tube, we call that acinar. Kind of forms a sac, like we see here with this one and this one. Okay. Then if it's got both tubular and acinar portions, like what we see down here, we call that a tubuloacinar. Okay. Now, we can also throw in terms like coiled and branched in the middle, but we'll really get into any of that specific stuff. Down at the bottom is a description of how we classify exocrine glands based on their function, which relates to how they release their secretion. And there's 3 functional classifications. The first shown here is illustrating a merocrine gland, like our salivary gland in our cheek. Key feature here is we produce a substance. Substance is transported to the Golgi, and then bubbles break away from the Golgi, called secretory vesicles, which then fuse with the plasma membrane, releasing the substance from the cell altogether. Okay. Most exocrine glands of the body are this type, which includes not only the salivary glands, but also those of your pancreas. Okay. Apocrine glands are very similar to merocrine, but differ in cell composition. What happens is we still do the same thing. We use the Golgi to create these little bubbles. The vesicles, instead of being released from the cell altogether, accumulate on one side of the cell. And then as you can see, that cell pinches inward and breaks into kind of two parts. The rest of the cell remains normal, and then the side where all those vesicles accumulated breaks away. And so the cell releases not only the substance, but also a portion of itself. And the remaining portion of the cell then can repair itself and repeat the process. And examples obviously shown here are your mammary glands in lactating females. Holocrine sweat gland, or excuse me, holocrine glands. We have holocrine sweat glands, no, we don't. I'm sorry, I'm totally talking wrong. Apocrine sweat glands, not holocrine. Holocrine glands are those that kind of the same thing. We create bubbles, vesicles, but instead of a portion of the cell pinching off, the entire cell actually dies and becomes part of that secretory process or product. Excuse me. This is what happens when we sweat. Or excuse me. Gosh, I did it again. When we release oil, what sebaceous glands secrete in our skin. I'm sorry, I can't talk apparently this afternoon. Okay, so I've already started to talk about some of these. Let's talk about the actual distinctive, very important skin glands. We'll begin with the most prominent ones are sweat glands. Fancy term for those are our sudoriferous glands. As I mentioned, most numerous glands of our skin produce a water perspiration that functions to cool our body. Find them over the entire skin surface except our nipples and parts of the external genitalia. And there's two types that exist. If I go back to that picture that was on that first page, okay, we can see them here in our picture. Okay, I had you guys highlight them and make sure you knew them. Okay. If you follow where the duct goes, that's going to be the key to telling them apart. Okay. The duct of an eccrine sweat gland opens up at a sweat pore, which is just an opening at our skin surface. Whereas with an apocrine sweat gland, they're always associated with hair because they open like a sebaceous gland into kind of the neck area, almost where the hair pops out of the skin. So that's for apocrine sweat glands. We'll talk a little bit more about those here. First, let's talk about eccrine. These are the primary sweat glands of the body. They're also known as merocrine, and that's because of this we just talked about, how they secrete. We find them abundantly on our palms, soles, and forehead, places that get super sweaty pretty easily, especially when we're hotter, nervous, that kind of stuff. They consist of a simple coiled tubular gland in which the secretory portion lies in the dermis. Duct extends to open in a funnel-shaped pore at the skin, as I was showing you on that first page. Sweat that's produced by these glands contains a lot of water. It's mostly water. It's also got ions like sodium and chloride, which together form salt. Most people are aware that they have salt in their sweat. Urea, uric acid, ammonia, amino acids, glucose, and lactic acid, which are a combination of different things. Apocrine sweat glands are simple coiled tubular glands. We find them where hairy. Armpits, groin, areola, bearded regions of the face in male adults. Within the secretory portion of the gland is down in the subcutaneous layer. The excretory duct opens up, and the key here, once again, is that they open up the hair follicles. They produce a different type of sweat that contains fatty acids, which bacteria tend to love. And bacteria utilize those, and in doing so, they give off an odor. And this is a fancy, basically, way of describing BO. Bromhidrosis is the condition, or the body odor, I should say, that causes the condition, BO. Sebaceous or oil glands, simple branched center glands, where the secretory or secreting portion of the gland lies in the dermis, usually opening into the neck of a hair follicle. We kind of already talked a little bit about that in this lecture. Find them all over the body except the palms and soles, which, if you think about it, have no hair, which should make sense. We don't have these glands. Also, on top of that, we wouldn't want oil being released in the palms of our hands, bottom of our feet, because that would make them slippery. and it would be harder to walk when we barefoot or to hold things without them slipping out of our fingers and hands. The oil that they produce is actually called sebum. It's an oily secretion made up of triglycerides, so that's a fat, cholesterol, and other fat, proteins, and inorganic salts. Function of it is to keep our skin and hair from becoming dry, brittle, and cracked. Acne is an inflammation of a sebaceous gland. That's what a pimple is. Ceruminous glands are modified sweat glands specific to our external ear. Secretion includes not only sebum, but dead epidermal cells, forming the fancy word cerumen, which is just a more complicated way to say earwax, and that's why they call them ceruminous glands. So, function of earwax is to provide a sticky barrier to foreign bodies and insects, keep the eardrum pliable, waterproof that auditory canal, and bactericidal effect means that it can actually kill some bacteria. Secretory portion of these glands lies in the subcutaneous layer. The excretory ducts open either directly onto the ear canal, or I really should say into the ear canal, or into ducts of sebaceous glands, which then open up into the ear canal. Mammary glands are the milk-producing glands that develop within the female breast only during pregnancy and lactation. They are modified apocrine sweat glands, producing a much richer secretion. And unlike sweat glands that each gland opens up into a different area, all the ducts of mammary glands kind of end up in the same place, opening up into the nipple for very efficient secretion, which is how the baby can get its nourishment for those first several months of life after they've been born. We'll talk about mammary glands. Actually, we have one slide, the very last slide we cover in this class when we talk about breasts, when we talk about the female reproductive system at the end of the semester. Okay, our final structures of the integumentary system are fingernails and toenails, which are scale-like modifications of the epidermis, forming a clear protective covering on the dorsal. Remember, dorsal and posterior refer to backside, surface of the finger toe. Composed of hard keratinized epidermal cells, and they protect the distal end of our digits, but they also enhance touch perception and manipulation, allow us to grasp small objects, and one of the best things, they're used to scratch and groom and very in the body, scratch and groom the body in various ways. And if you can't ever itch a scratch, you know how awful and annoying that is. So fingernails are great for that. Okay, structurally, finger and toenails are pretty complicated. The first structure I want to talk about is the nail body. The nail body is the portion of the nail that you can see makes up the majority of it. Can be described as the visible portion of the nail, comparable to the stratum corneum of our epidermis, with the exception it's flattened. Keratinized cells are filled with a harder type of keratin, and we're not shedding off the cells like we were at the corneum. At the very end of the nail body is the free edge, so it's the distal end. Depending upon how short you keep your finger and toenails clipped, this is what you're taking off. So depends on, and that's why it says may extend past. If you cut it short, it might not. It's white in appearance because of no underlying capillaries. And on the other end of your nail, the proximal side, is a kind of quarter moon crescent-shaped lunula, another white area, which appears white due to a thickened region of the epithelium in that area. Actually, internal to that, which you can only see if you have like a sagittal section, which is what's shown here, would be the nail root, which is embedded in our skin in the finger or toe, depending on what we're looking at. In this case, the finger. Okay. Right there where the nail root is also happens to be kind of like the hair root, a nail matrix, which is where cell division takes place, just like what we saw with hair, where the hair matrix went through cell division, and this is what causes your nails to grow. Okay, so it contains dividing cells, produce new nail cells. Okay, let's talk about the hyponychium. Hyponychium, fun word to say, is shown here in our picture is a thickened region of the stratum corneum, and it's actually beneath the free edge. So it's right there beneath the nail body, okay? And specifically more so the free edge than the body, because the nail bed is beneath the nail plate, okay, extending from the lunula to the hyponychium. I already talked about the nail matrix. The aponychium, or cuticles, our last structure to talk about. It's this little band you can see just proximal to your fingernail and toenail. It's a narrow band of epidermis extending from and adhering to the lateral border of the nail wall. Occupies the proximal border of the nail and consists of stratum corneum. Down here at the bottom is a actual cadaver finger, I believe, yeah, fingertip, it says. Looking at it under a microscope 5 times, basically showing the exact same thing as this after a section has been taken along that sagittal plane, where we can see, fairly well anyway, the different parts of the fingernail and the finger associated with it. Not going to worry about that one, but this would be in this one as well. Pictures to know. Okay, so that is the remainder of our lecture on the integumentary system. Thanks.